We have with us Assistant Professor of Biology in the WM Keck Science Department, Sarah Budischak. This is her third year with the Keck Science Department. She'll discuss why new diseases emerge in human populations and the role human activities play. Where did COVID-19 come from? How are scientists trying to predict the next pandemic? We are so excited that all of you are here, and she has promised that no science degree is required. There'll be lots of great background and information. Professor Budischak, thank you so much for being here. The show is yours.
All right, well, thank you so much, Evan, and thank you all for being here. It's great to see you, and I'm so excited that you're interested in this topic and trying to more deeply understand the current pandemic, as well as what will likely be the next one. So I study disease ecology and evolution, and I've been trying to tell my parents for years what this field is and why it's important. And the current pandemic just really is an excellent, it's awful, but it's a good example of why we need more research in this sort of field so we can try to mitigate and minimize the consequences of the next pandemic, as well as the one we're currently in. So I'm really happy to take questions as we go along as well. So I'll try to keep an eye on the chat. You can also use the raise your hand function, or if you're not in an environment where it's easy to like click the mouse buttons, you can go ahead and just speak up. I'm very happy to be interrupted. So here's where we'll start. Okay, so before we start, I just want to make sure we are all on the same page in terms of terminology, so the words I use are clear and understandable. So what is an epidemic? Well, first, to figure out what an epidemic is, we have to contrast it to an endemic disease. An endemic disease is just constantly present, kind of very little fluctuations, is around all the time. Something like malaria would be an endemic disease. Whereas an epidemic is a disease that suddenly has a rise in new cases, and you get this big increase in cases over time. So you get this kind of wave shape if you look at the number of people who are sick over time. And a pandemic is just a large scale epidemic. So one that happens in multiple places or to a large number of individuals at the same time. So how do epidemics start? The term we use for this is called disease emergence. So these diseases come from somewhere or change in some way. So there are three different ways that diseases can emerge or start to become a problem. The first is that they can change in their geographic range and spread to a new area. And often when they do this, there's a lot of new hosts. Hosts are the individuals that get infected, whether it's animal or human or plant. We have that kind of catch-all term for whoever's getting infected. And the Zika pandemic or epidemic a couple years ago is a good example of this. Zika is caused by a virus that's transmitted by mosquitoes, and it is not particularly lethal. Only about 1 in 5 people become symptomatic. They're sick for about a week, and it was usually a mild illness, but it was also linked to really severe birth defects. And this epidemic occurred because it was able to be moved from areas in Southeast Asia to the Americas, into South America, and there it spread. It found all these new individuals, lots of new mosquitoes to infect, and that's what caused the epidemic. It got moved from place to place. And if you look at the number of cases, it follows that exact characteristic epidemic curve where you get this big peak and then it trails off over time as people recover, as public health measures are put in place, things like controlling mosquitoes. People become immune, you can get to herd immunity. So there's a lot of reasons why these epidemics trail off. The second way that diseases can emerge is if they change in virulence. And virulence is how costly they are to their host. It could be in terms of death or just how sick individuals get. And there's kind of two ways that this can happen. It can happen because of a current infection that suddenly becomes resistant to antibiotics or a vaccine. and then doesn't work anymore. It can also happen when some sort of infection just mutates, and for whatever reason gets that random mutation happens to be one that makes it replicate more in a host or more deadly to a host. And tuberculosis is an example of a disease that's emerging because of resistance. Drug-resistant tuberculosis strains are spreading globally, and they are leading to this resurgence of the number of tuberculosis cases that we see in people. And then the third way that diseases can emerge, which is the one we're going to spend the most time on today, is when they increase in something called host range, which just means that they can jump from one host species to another. For example, from like a primate to a human or a bat to a human. And this can be directly from a wildlife. Oftentimes there's some sort of domestic animal that it spreads from. So if you've seen the movie Contagion, you know that Gwyneth Paltrow gets infected with a new virus because a bat pooped on a pig, which then got fed to her and she got sick. Well, that actually really happens. That is a legitimate way that these diseases spread into humans. And sometimes these diseases can then spread from human to human, and that's when they really cause problems. And especially now that we have airplanes, a sick human can get to the other side of the world really quickly. And so that this host range can go to increased geographic range really quick because humans are so mobile. And I think it's really important to stop here and note and think about for a second that about 75% of emerging infectious diseases come from non-human animals. So we can't just focus on human diseases if we're going to understand these. We really need to be studying diseases in wildlife to understand their properties, to know what's out there, what they do, how to treat them, how to study them, because they are such a big risk to human populations. So one of the questions a lot of people in my field study is what kind of animals have the potential for these diseases that jump to humans? And we have a special word for that called zoonotic. It just means that they can spread from wildlife to humans. So often animals that have lots of contact with humans spread diseases to humans. That makes sense. The ones that are closely related to humans evolutionarily are also more likely to spread diseases to humans because to a bacteria or virus, we don't look as different from their original host. So our immune systems, our cells, the receptors we have on those cells that they use to get in, a lot of that is more similar in species that are more closely related. And then the third category that I have here is that some animals just have lots of jumpy infections that can spread to lots of different species. So this is an example of a research study in my field. This is from 2017. So this is before all this started. People are really interested in how likely are viruses to jump to humans. And this graph has a lot of stuff going on. So I'm going to walk you through it. Let me get my laser pointer out. Okay, so down the middle, we have nice pictures of different kinds of animals. Things like bats, primates, rodents. These are right here. This one's ungulates, that's all hooved mammals. All right, so we have different kinds of animals. And then what proportion of the viruses that we know they have also infect humans? So the ones we worry about are the ones where their averages are really high. So this black bar in the middle of the gray box, or over here, it's on the side of the box, they're showing that these kind of species, these ones at the top, have a lot of species of viruses that jump to humans. All right, so these may be the ones we should be more careful about and focusing our research on. On the right, this one talks about total virus richness. So how many viruses do we know that they have? And when you look at this side, you see that these ones here at the top actually only have like 4 or 5 dots. So basically the take home point is we only know about the viruses in armadillos that infect humans. We haven't studied armadillo viruses just for the sake of studying armadillo viruses. We only know the couple ones that also infect humans. So up here, we really haven't sampled enough. They're probably pretty high just because the only ones we know about are the ones that also infect humans. They're the only ones we're testing for, looking for. So the ones that have a lot of viruses that spread to humans that we've actually studied well enough are the bats, the primates, and the rodents. And evolutionarily, bats and rodents are the two closest groups of species to humans. So that evolutionary relatedness is really showing up here. The other thing that's interesting on this graph are these kind of burgundy dots. All those are domestic animals. So we have studied species that are our pets, our farm animals, a lot more. And indeed, some of those also have viruses that are really likely to jump to humans. We can also study where spillover is happening, or this is another way that they looked at to see what kind of species have viruses that spill over to humans. And they broke it up by where they're found geographically. So the more purple colors on these maps are the species that have viruses that can spread to humans, and the yellow are fewer. And you can see that in Central and South America, there's a lot of bat diseases that can spill over to humans, as well as in Southeast Asia, whereas rodent infections that can spill over to humans are much more common in Europe and in North America. So we can start seeing these patterns and helping focus our research, because there's so much wildlife and so many viruses and bacteria in wildlife. We need to know what to focus on. We can't just study it all. We can also study where we need to know more. So using what we know and kind of inferring patterns that of how many new species we're discovering and things like that, we can figure out where are these viruses that we don't know about. And we can look at that and tell us where to start sampling wildlife. You can see that a lot of the same patterns are here, but there's also some different ones. So we kind of know about these rodent-borne infections in North America and Europe, but we haven't studied them as much in South America. So it can really help us figure out where to be studying these viruses. One that has come up a lot on these maps and these darkest colors of where there are viruses and where there are missing viruses are in the bats. So bats are a key reservoir. So they're this key place where viruses live. They have lots and lots of viruses. They also seem to have viruses that make other species really, really sick, but don't have as strong effects on the bats. And in fact, a lot of the most recent epidemics in human populations have come from these bat-borne viruses. One of those is Nipah virus, as well as Hendra virus. And in these cases, these viruses from bats spilled over into domestic animals like horses or pigs, and then got spread to humans. And sometimes they go right from the bats to humans. So these bats not only seem to have a lot of viruses, they have a lot of deadly viruses, and their viruses seem to be able to spill over to humans fairly easily. A lot of times, these same viruses spill over not just one time, but multiple times in multiple different places. And indeed, both the past SARS coronavirus epidemic and the current pandemic are the most likely animal reservoir for this virus, where it started before it jumped to human populations, was bats. So this is a phylogenetic tree. This is how we study where these viruses come from. And you can think of it as like a family tree of who's related to who, branches that are side by side are more like siblings. If you go from one to another, they're distant cousins. And it can give you an idea of which samples of virus are most closely related to each other. So let me get my pointer out again. Over here, this is like a long time, like think of it, this is back in time, and then the virus is changing, and each time it has offspring, they're a little bit different. And we've sampled some of these offspring viruses in different species. The samples from bats are marked in blue, and the samples from humans are marked in red. So you can see that the sort of oldest samples are all from bats. And SARS coronavirus, the first one that spilled over to humans in 2003 and started a big epidemic, that spilled over from bats to humans. We don't know if there was some other animal in between, like with Nipah, there could have been a pig, a civet cat, any other animal. It could have gone directly from human to bat or through some other animal. But where it originally started was in bats. And we see that same thing for the current pandemic, where bats are kind of the oldest ones. They're more the ones on here. We've sampled some pangolins, and they're kind of distant cousins to it. But it's more likely that bats just spread the virus to pangolins. And it's very unlikely that pangolins were the original source of this virus. And then once it gets into humans, and when it spreads, it can start mutating and accumulating mutations and becoming different. And what we see now is that some of these mutations are helping it spread in human populations and becoming more and more common over time. All right. So I think it's really important as we think about these emerging infectious diseases to realize that this hasn't always been the case. There haven't always been this many. I've talked about several different epidemics that have happened just in the last 10, 15 years. And a little bit of this is that we are getting better at finding them. But it's also overwhelmingly true that there's more of these new diseases emerging. This is an example of some like the best data that we have. This is a study that was done in China, and it's complicated. So let me walk you through these axes. So on the x-axis here, this horizontal one going across, we have year. And then on this vertical one, we have number of provinces affected. So think about it as how wide did these infections spread? Each red dot is an emerging infectious disease, and how big the dots are tells you how many people got sick. And what you notice is there's more red dots and also more larger red dots as you move towards the present. And this doesn't even have COVID-19 on it, which would be way up here because it's spread everywhere and has infected many people. All right, so there's a quick question. Let's see. So I see a really great question in the chat of why do bats get so many diseases? This is one that people in my field are really working on studying. Part of the, a lot of the hypotheses have to do with the fact that bats are the only animals that fly. And when flying is such an energetically costly activity, so think about it, if you run really hard, your muscles are sore, there's lactic acid, there's damage, and anytime you just use a lot of energy, it can cause damage to cells. So bats, because they fly, have had to evolve mechanisms to get really good at cleaning up that damage. It's due to something called free radicals, and they're really good at cleaning that up, and really good at dealing with cells that are stressed and taxed, and sort of removing them without causing too strong of an immune response. It's just something normal. They have to deal with all these cells that get damaged. And the thought is that, or one hypothesis is that these viruses, they cause similar damage to cells. And bats are able to kind of clean up that damage and live with the virus without having these sort of immune responses that are overzealous and cause a lot of health problems. A lot of the mortality due to COVID is due to immune responses that get elevated too strongly. And bats, because they're used to all this damage from flying that's similar to viruses, they're also really good at cleaning up this virus damage without it causing big health problems. So that's a main hypothesis. Another, but it's one that's being debated right now. There've been conflicting papers coming out saying, well, there's just a lot of bat species, so we see a lot of bat viruses just because there are a lot of bats. So numerically, it might just be because there are a lot of bats and they're very similar, but I think there's also some just different physiology in the bats that helps them survive with so many of these viruses. Like think about rabies. Rabies kills every other kind of mammal besides bats rather quickly, but bats are able to be this host for rabies for a long time. And it does eventually kill a lot of bats, but they're able to sort of live with these viruses for a long time. So yeah, it's something we're still working on figuring out, but there's a lot of interesting hypotheses that we're getting more evidence to support. All right, so saw this pattern. Emerging infectious diseases are increasing, and the overwhelming evidence suggests that human activities are increasing these chances of emerging infectious diseases. So just having lots more humans around, more livestock at high density, more domestic animals, this increases contacts between individuals and makes disease spread easier. Humans are also increasing the movement, not just of themselves, but of other animals all over the planet. So the first SARS epidemic, it spread to, what was it, 26 countries? And it spread to like many countries just within a week because humans are so mobile. But livestock, movement of animals for the pet trade is also increasing these risks. And often when animals are being shipped around the world, they're in really dense, stressed conditions. So it's easier for a virus that to have the opportunity to jump host species, as well as to replicate more because stressed animals have more weakened immune defenses. So it's just kind of really good conditions for the spread of infectious diseases. Climate change is another. This is especially important for things like Zika and any infection that's spread by mosquitoes. As temperatures increase and rainfall patterns change, there's now new areas that where these vectors can live, as well as a lot of times there's now a longer time period in the year where mosquitoes can be out and active and spreading infections. There's good evidence about things like urbanization and deforestation playing a big role in the emergence of these infections. In Nipah virus, deforestation has been linked to the spread of these bat viruses into human populations. And also the overuse of antibiotics. So not all of these infections, these emerging diseases are coming from wildlife. A good number and an increasing number are coming due to the overuse of things like antibiotics and getting antibiotic resistant strains of infections that are already around, things like MRSA and drug resistant tuberculosis. And this is a map of where these diseases have emerged. This is a dated one, but it's kind of the best. There's no updated version of this study. And it also is going to be sampling bias. We don't know what we haven't tested for, but a lot of disease emergence is happening in the Northern Hemisphere. And a lot of this is due to this overuse of antibiotics. And a lot of, often foodborne infections, things like that. Also, just international travel, people picking up infections and they may get tested for in a different place. I wanted to go back and show you the actual evidence to back up that land use change example that I had in my list. This is a study that looked at what are the factors that are related to emerging infectious disease risk. And what they found, there were several things that were really important for predicting where diseases would emerge. They emerged where the more people, the more likely there was for a disease to emerge. Also, if you had more mammal biodiversity, so more wildlife species, plus more people, okay, we're setting the stage. And then also land use changes. And if you had these three things going on together, that's where you saw the highest risk for emerging infectious diseases. And the COVID-19 pandemic, it emerged here in kind of central Western China, bright yellow on this map. It was not at all surprising to me that the current pandemic was a bat-borne virus from this region of the world, because like this is what we were predicting. And we also had scientists there studying these things and trying to understand them and learn about them before it happened. I can skip this one. It also wasn't surprising that it was a coronavirus. Coronaviruses often jump host species. This is another phylogenetic tree of different samples of coronaviruses. This red one is humans, the current pandemic strain. And you can see, like in this family tree, it has spread to lots of different species, like mink and ferrets, humans, bats, different kinds of bats, and camels. So these coronaviruses often jump host species, but it's just very rarely that they actually get further than just that first case or two.
Sarah,
but sometimes they do.
A follow-up question
Yes.
just came in. What do you mean by land use change?
Oh yeah, okay. So what I mean by that is there's lots of different ways to study this. One of the ways they do it is look at satellite images and say, over time, has what was in place in an area changed? So often it is cutting down forests and for towns or for fields, for pastures. So they're taking these satellite images and saying, has this area changed in what it looks like over time? Has it become more developed? It could become more forested, but almost all the changes are going in that way. So this is a breakdown of some of the things they looked at. And you can see like cultivated or managed vegetation, whether pasture increased or not, whether it's basically, they just take these things and they can see whether it's bright green trees or if it's been developed in different ways. Yeah, great question. So these coronaviruses are out there. We know they're kind of jumpy viruses, and they can do this because the pool of viruses that are in bats are really diverse. So this was a study that was published in 2017, and they went out and sampled all these bats to look at coronaviruses in bats in 2017 in China to study them because they were afraid that this might happen. And what they found was that, so I'll tell you the punchline and then I'll talk you through this graph. The punchline is that different bats had different variants of this virus. So this virus wasn't just one thing, but it had a lot of different versions of itself circulating in bats. And what that means is there's this increased chance that one of those, if there's a lot of different types, if it's really unlikely it can get to humans or spread from human to human, well, the more variants it has, the more chances it has that one of those mutations is going to actually help it spread from human to human. So now let's look at this graph and try to get there from it. So all these rainbow colors are different samples of virus from different individual bats. So each bat has its own color. And then the whole length of this is the genome of this virus. So all the genes that this virus has. And this virus has genes that do different things. These first two ORFs are just kind of the housekeeping genes. That's how this virus replicates. this S is the spike protein that you may have heard about in news articles. It is what sticks out of the virus, and it's what binds to a receptor on human cells and helps it get in. It is also, because it's sticking out of this virus, what your immune system can recognize to tell that it's a virus. So when you make antibodies, the antibodies are made to match that spike protein, and they label it, and then it gets targeted for destruction. The vaccines that are out are mimicking regions of this spike protein because it is this thing that our immune systems can see. It sticks out of the virus. And it's also the same area where this virus has lots of different kinds. And this is what kind of sets it up to be ready to, one of these might match the receptor in humans really well. So because they have all this variation there, there's more chances that one of these mutations will just happen to also work for a human receptor and let it into human cells instead of just bat cells. All right, and so like this is something we've been studying, we've been worrying about these. This is another study, this was published in September 2019, looking at the role of human-animal interactions and that coronavirus spillover potential in southern China. And what they found was that there were certain things on their survey that were made it more likely that individuals had antibodies to a coronavirus. So a lot of individuals they found had already been exposed to coronaviruses, not this current strain, but a different one of these other, or one that we didn't sample, one of these different rainbow variants of coronavirus these people had been exposed to them before. And what they found were big predictors of whether or not people had been exposed to coronaviruses was if they'd eaten raw or undercooked carnivores. So that direct contact with wildlife that could have viruses. Undercooked means that the heat hasn't killed the viruses. If they slaughter poultry, low income, if they had bats in their house, if they had rodents in their house, if they were lower income, and some of these things may be correlated with each other, if they handled live poultry. So all of these ways that humans are interacting with animals is really increase the likelihood that they had been exposed to coronaviruses. So human activities are tied to exposure risk and the spillover risk. Kind of my take-home message is that these coronaviruses, there is evidence of lots of spillover to humans. Like not all of them turn into pandemics. It infects lots of people regularly without turning into a pandemic. But the more times that happens, more likely one of those is just to hit the jackpot and actually be able to spread human to human. And this likelihood of spillover is related to particular kinds of human-animal contact. These bats are the primary reservoir for these viruses, and they have lots of different variants, which is increasing that risk that one of them will just by chance be able to spread in humans. COVID-19 is actually the third epidemic caused by a different kind of bat coronavirus. There was SARS, and then MERS, and now COVID-19. So every 4 to 12 years, there's been a new one spreading into human populations. So I'd say, based on this evidence, it seems like it's just a matter of time until another one spills over and is able to transmit human to human. But there are things we could do to minimize these risks, to increase monitoring in these areas that we know may have lots of viruses that can jump to humans, to change some of these livestock practices, to put research into how to develop these vaccines really quickly. It's been impressive how fast we've been able to develop vaccines to COVID-19, and those should be really useful tools And the more that we can get those sort of technologies and detection systems and research in these areas where there can be spillover, I think is going to really help make the next epidemic a much, much smaller one. All right.
Sarah, quick question that came into the chat. Why do you think there was such a gap? You know, SARS and MERS were only 4 years apart, and then there was a 12-year gap to today. Why do you think there was those two were so close, and then there was such a big gap?
Yeah, so mutations are random events. They just are kind of byproducts of mistakes of copying the virus genes. Each time it divides, some mistakes can happen. So these things are just happening randomly. So chances are, most of them are actually bad for the virus. Some of them are neutral and don't really do anything for it. And some of them are actually beneficial. And what seems to be the case in a lot of these is that the virus already has to have some sort of way. So even if there is a bat virus that has a mutation that would make it really able to spread in humans, chances are that bat virus is never going to meet a human. So it just there's all these sort of low probability events that have to happen at the same time for these spillovers to happen. So that makes them really hard to predict. So the virus has to have some mutation that it lets it infect humans, also let it spread from human to human. And that trait isn't something that's necessarily going to be beneficial while it's in the bat. So probably, even if it popped up, it'd probably go away because a virus spreading from bat to bat doesn't need to have this human to human transmission adaptation. So it's just like, yeah, these low probability events, but the more chances that we give it to happen, the more frequently they're going to be occurring. So the more people are in contact with bats, the more that we have livestock right up at the edge of forests at high densities. So we stress livestock right next to wildlife, that increases these chances. And especially if these viruses can go from a bat into some sort of domestic animal that then humans have a lot more contact with, just is more chances for this to happen. So it's just really hard to predict because like there's this random process and then randomly which bat happens to pee on some swine food. Like it's really hard to predict. But in the next 20 years, I would say we probably will have another one for sure. But yeah, it's really hard to predict because they are just, yeah, they're just like these random chance events. And then if the stars align, they can spread really rapidly. And the high density of people and the movement of people, once it gets into human populations, these infections are able to spread much further and more rapidly and infect many more people than ever before. So something like the first SARS epidemic, if that had happened 20 years before, it probably wouldn't have made it to all over the world. So yeah, it's just, I don't know, it's very hard to predict. We are trying to do this. There's lots of research out there trying to predict what the next one is going to be. The last couple of papers and research I showed you about were from this EcoHealth Alliance group supported by USAID Predict, and their partner labs in China that are really working on studying these viruses in bats. So yeah, it's really hard to predict because they're just so like these are unpredictable mutations and unpredictable chances where humans and bats and other animals are interacting.
So Sarah, so it's fascinating that I think we all remember the SARS and the MERS pandemic, but it feels like it was so foreign, so distant, so far away. And this one feels so unbelievably close. Mm-hmm. What do you think? Was it about the international travel, the higher density of the Earth now? Like, why is this one so much worse than the other two? Mm-hmm. Or is it? Maybe just have better detective devices.
Counterintuitively, this one is so much worse because it has a much lower mortality rate. I think MERS mortality rate is around 70%, SARS was about 40%. And when there are mortalities, diseases get studied. When people just start dying for unexplained reasons, there's a lot of research. And we can do that contact tracing early and figure out what's going on. And when they have these high mortality rates, generally everyone who's sick shows symptoms. So so these diseases could be noticed, they could be controlled, they could be quarantined. People, like if it's a disease that has that high mortality rate, you go to the hospital even if you feel a little bit sick, or even if you've had maybe slight contact with someone that was known to be infected. But when you have a disease that has a much lower mortality rate, plus one where a lot of individuals who get it don't show symptoms and don't feel sick, it can spread without people noticing. And it just makes it much, much harder to control. So that is why this current pandemic has been able to spread much further and is much harder to control, is because it has this lower mortality rate and so many individuals who are infectious without showing symptoms. All right. Cool. So that's all I had in terms of slides, but I'd love to field more questions if you have any.
Well, thank you very much, Professor Budischak. So we do have some questions that came in the chat. I do ask anyone, if you do have a question, put it in the chat or click raise your hand in the reactions tab. So a question came in about Zika. Why did the science community decide to not create a vaccine for the Zika virus?
Oh, that is a great question. So I think they might be working on it, but also vaccines, when you have infections that are spread by mosquitoes, their transmission is a bit different mathematically. So a mosquito is only going to bite so many individuals before it dies. So let's say a mosquito bites 5 individuals. That mosquito is going to bite 5 individuals, whether there's 5 individuals in that room or 50,000 individuals in that room. Whereas a respiratory infection, one that's you get by inhaling it, if there's 5 people in that room, 5 of them might get sick. If there's 5,000, they might all get the infection. So we call that density dependent, where the more individuals there are, the faster a disease can spread. So that's what's true about the current pandemic. And because of that, there's a density where it's too low, where it's let's say that virus only infects 1 in 4 individuals a person has contact with. If they're only around 1 susceptible individual, the virus may not spread, and then it kind of dies off there. And you can have these chains of transmission break when there's too few susceptible individuals. And that is what the principle of herd immunity is about. So we are trying to get enough individuals vaccinated that we can stop these chains of virus transmission. And if enough of them are stopped, if the virus just bumps into enough resistant individuals, it will just stop spreading. We don't have to vaccinate everybody, but if we vaccinate enough, we can stop transmission enough that it will really slow, and then the spread will just die off. So that's what we're going for with the current pandemic. With something like Zika, that mosquito, it can still spread just as well when there's few individuals as many individuals. So you're never going to get to this point of herd immunity. So vaccination is a really important tool for reducing the health consequences of that infection, but it is not going to be able to get herd immunity and protect entire populations the way that we're going for with COVID-19. The other thing is that cases really died off after that big epidemic. So when you have places that have limited funding, if it's not the biggest problem at the moment, there's not as much investment. If a disease is elsewhere and not in the countries that have the most money, less investment. So there's kind of both this economic piece of it and also this kind of biology of the disease, that these mosquito transmitted infections just are different, and we're never going to get to herd immunity with them.
Thank you. Sue Ann wrote in the chat, does COVID-19 also have a longer incubation period than other viruses?
So with all viruses, one of the things that makes them harder to control is if people or any animal that has them can transmit it before they show symptoms. And the longer individuals can transmit before they show symptoms, so kind of during that incubation period from when they get infected and when they show symptoms, if they can be transmitting in there, and the more they can be transmitting in there, the harder disease is going to be to contain. I don't think this incubation period is particularly long, but what is unfortunate about it is that people are infectious way before they show symptoms. And this is true for a lot of infections like flu and there's a lot of viral infections where people are infectious before they show symptoms. But it just makes them much harder to control.
How worried should we be about new COVID strains?
So there's some promising information that the vaccines seem to be doing a good job so far of offering protection, especially against severe infection, even of the newest and most different strains. The more chances we give the virus to replicate, the more different it can get and the more chances it has to kind of get around our vaccines. So as we're ramping up vaccine distribution and reducing cases, all of that should also reduce the likelihood that there are new strains that can get around our vaccines. The more we use the vaccines, it seems a little counterintuitive, it's a bit different than antibiotics. The fewer chances we're giving this virus to evade them. So that is really helpful. So far the evidence seems to be that reinfection is really unlikely. So there have been instances, but it's a handful of known cases where people have gotten a strain that's different enough to get infected a second time. So that seems to be really rare. So that's a good sign. Another one is that when we make antibody defenses, even though these vaccines just have kind of one variant in them to train our immune systems, as we develop antibody responses, we also make we don't make all antibodies that are exactly the same. We kind of make this little family of B cells that are producing really similar antibodies. So even if we get exposed to a second strain that's not exactly the vaccine one, there's a chance that maybe one of the other sort of closely related ones, when they got that signal to start making antibodies, some of the ones that are just a little bit different might be productive as well. And the other thing that I think is really hopeful is that because of the technology that Pfizer and Moderna are using in their vaccines, it would be really, really easy to change what's in the vaccine slightly to offer protection against a different strain that might emerge later. So I think there are going to be variants and there may be some cases of reinfection, but I think we have a lot of tools that none of it is going to be as bad as what we've experienced so far.
So you mentioned tools, and we actually had a question in the chat from Jose about mitigation tools. Mm-hmm. What is the best way to mitigate the spread? Is it a total lockdown like what China did with Wuhan? Mm-hmm. Is it what we did in California of safer at home? Pre-vaccine? Is it masks and hand washing? What is best to contain something like this?
It depends where we are at in the stage of infection. If it's so the ideal thing to do is, as soon as it's known, to do quarantining and isolation of all known cases, and around areas where there's more of those, to put in public health measures like distancing and hand washing, to wearing masks to reduce transmission. Once it's at the community level, once it's just kind of spreading and you can't even do contact tracing because you have no idea who you got it from, because anybody could be sick. That's when these broader public health measures like social distancing, stay at home, face masks, that's when those pieces are really critical, because there's just so much transmission. We can't get to the stage where we can do the effective thing because there's just too much transmission. So the more we can reduce that, the more we can reduce contacts, and especially people who, if there are contacts, like there are critical services that need to be done, reasons why people need to be out of their homes. But if we can reduce as much as possible the things that maybe aren't as critical, like I could work from my house here. And I mean, if we're all dying to get back to campus, and I'm sure your students are as well, but we can make it work for the time being. Whereas people who work at like the electrical grid, we need them there. So I think if everybody can just do as much as they can to reduce transmission, that's the most effective thing at this point. Plus vaccines are going to be amazing.
We have time for one more question. It goes to Tim Galbraith in New York. COVID vaccine development was inside 12 months, setting a record. Is that now the new benchmark, and future vaccines will roll out with the same speed or faster?
I believe so. So one of the reasons we were able to do this so quickly was people had been working on this mRNA vaccine technology for other infections. So this is a vaccine where they basically can just compose the string of letters in the genetic code that's in there and make this little molecule. They can design it however they want. So they were working on it for a totally, I forget if it was HIV vaccine, for some other type of thing. And they were able to just change out what was in that vaccine. They had all the technology in place just to substitute in these different strains. And what they found was these kinds of vaccines can actually be safe and effective, which were like really critical pieces to find out. So now we have this tool that we can change really easily, that so far, like for these infections, seems both safe and effective. So I think it's going to be much faster to develop vaccines. I mean, we still have to go through the different stages of human trials to make sure they're both safe and effective each time. But the starting point, we're going to start with something that is really easy to change and like can be changed in a couple of, like, I don't know, within a month. Like you can have something new and ready to be tested. So yeah, I think it's really, really promising technology.
Great. Well, Professor Budischak, thank you so much for taking your time on a Saturday morning. We really appreciate it. We love seeing a glimpse into what our students are learning and how they're learning and who they're learning from. So thank you very much. And thanks to everybody for spending an hour with us this morning. We hope to see you at programs next week and the week after. A friendly reminder that the wine tasting with President Hiram Chodosh and Priya Junar, cutoff for registration is Monday. So if you're going to join us for the wine tasting, please register by Monday. Otherwise, we have a wonderful program on Thursday about off-campus study in our Silicon Valley and DC programs, and more program the following week. So do, Jeannie, if you put the URL in again to future family programming for Spring Family Connections, that'd be great. Until then, everyone, have a great night, a great day, a great weekend. Thanks so much. Feel free to unmute, say hello, say goodbye. We'll see you all later.
Thank you all for coming.
Thank you, Professor. That was great.